Okay, so let's get started. I won't take a lot of time to be in the introduction here. I've said some of these things before, but let me begin by just mentioning again, welcome to this lifelong learners class on the ecology of infectious disease. I want to remind you of what ecology is, the study of the distribution and abundance of species. And in all cases that I'm aware of, communicable diseases or infectious diseases involve some other species. So by definition, if we're interested in disease and infectious disease, we're interested in ecology. It's important. It is fundamentally important to ecology. We have really a gift of having four terrific speakers in this class, and today's speaker, Dr. Rick Ostfeld, with the there we go. The Institute of Ecosystem Studies, the Cary Institute of Ecosystem Studies, is undeniably the world's expert on this subject. I'll just say a little bit about Rick, not too much, but I met him as an undergraduate at UC Santa Cruz. He did his bachelor's here back in the 70s sometime when I first came here. I think I met him in 78 or 79. We actually met at a softball game, as I recall. And we've been friends ever since. We interacted a fair amount when he was an undergraduate. He was actually working on sea otters, and then he went on to Berkeley, did his PhD at UC Berkeley, and eventually wound up on the East Coast. And in this position with a research institution, he's a little different from most scientists. Most of the scientists we hear from have direct affiliations with academic institutions. Rick has been with the Cary Institute virtually his entire career. And has grown into a disease ecologist more or less from about that time. Became interested in forest ecology, the ecology of small mammals. He was trained in mammalogy, and then really became interested in community ecology and disease ecology, because I think largely because Lyme was such an important part of the system that they were working in there. And he's been there ever since. He's had a very strong impact on the field of ecology and on disease ecology in general. One of the very best. He's done some beautiful work on Lyme. The thing about Lyme that to me is so interesting is that it brings out some of the complexities of ecology of disease. Many of the other diseases that we hear about, we don't really know as much about that complexity, and Lyme is fascinating for that reason, as well as the fact that it is simply a disease that we're all concerned about. Even here on the West Coast, we're concerned about disease, but if you lived in the Midwest or especially in the Northeast, you would be extremely concerned about it. So it is a disease that is highly relevant to human welfare, human behavior, and everything about our lives. So I think with that, I will simply turn over to Rick, ask him to screen share, and we'll get started with his lecture. And we'll do this more or less as we have. That is that he'll speak for a while, and then he'll stop at a convenient point for people to take a little break, and then we'll come back and finish up. So join me remotely in welcoming Rick. I'm so very pleased to have him, and I know this is going to be a very informative talk. So Rick, please go ahead. Thank you.
Okay, so
Jerry, you can go to the top, and yeah, good.
So I have just, at least I think I'm sharing my screen. I just want to make sure that I am. And just to check on whether you can see my cursor moving back and forth on the word ecology.
Yep.
It's wonderful.
Working great, Rick, and I see the screen. Everything's fine. I think everyone else is muted. So
okay. Yeah. Great. Okay, wonderful. Well, thank you, Jim, for that that kind introduction. It's a pleasure to be here as a researcher at a private, not-for-profit research institution without a regular teaching gig. It's a particular pleasure to be able to teach a class. So I very much appreciate being invited to do so. And I will be pausing partway through, and I thought that could be a time for interim questions and followed by a short break. And then I'll launch back into more slides. And I'll also try to keep track of time as I go. So I want to start by placing this talk on the ecology of Lyme disease in a somewhat broader context. That is, if my, let's see. I have to figure out how to advance. There we go. It advanced on my screen. Did it advance?
Yep.
Okay, good. Okay, so this is a slide from a paper that's now about 12 years old that is on the emerging infectious diseases of humans that have been documented somewhere on the planet since about 1940, up until shortly after the turn of the 21st century. And there are a couple of things to note. I guess one thing is that 2 weeks ago, the topic was zoonotic disease. So I won't repeat what a zoonosis is in any detail, except these are diseases that humans catch from pathogens that reside in non-human vertebrates. So we get zoonotic diseases from non-human vertebrate animals, typically mammals or birds. And so that was the topic 2 weeks ago. Last week, you went into the ocean, as I understand it. I wasn't participating in it. And this week, we are moving onto the land to talk more about a particular zoonotic disease, that is of Lyme disease. So that's, and I guess a broader context is that none of you need any convincing that we live in an age of emerging infectious diseases. Since the COVID pandemic has broken almost exactly a year ago today, we have been under the onslaught of the latest of these emerging infectious diseases. So just to remind you about what that means, an emerging infectious disease of humans is defined as either one that is brand new, it was never known to exist and was recently discovered. And that is the case with SARS coronavirus 2, the causative agent of COVID-19. Or it could have appeared suddenly in a new place where it was known to occur in some place for a while, and suddenly it appeared elsewhere. That would be the case with Zika virus, which jumped several years ago from the old world to the Western hemisphere. Or it could be a disease that's been known for a while and it suddenly gets much worse and starts sickening or killing people in much larger numbers. So an example of that would be Ebola virus, which has shown a series of different emergence events where it's suddenly gotten much worse. Lyme disease, that I'm going to be talking about today, is one of these emerging infectious diseases, and it arose in the northeastern United States, as I'll describe, back in the late 70s, early 80s. Note how many of these emerging infectious diseases have been described by scientists, 335 over a several decade period, and this is grossly undercounting the number that have emerged since 2004, and of course their impact. So as I said, Lyme disease is one of these emerging infectious diseases of humans. It was first discovered in the late 1970s and fully described in terms of what the causative agent was by the early 1980s. And shortly thereafter, it became a notifiable disease, which meant that the CDC requires state health departments to report annually on the number of cases that occur in that state. And what this slide demonstrates is that from the early 1980s, when there were a few hundred cases per year, to early in the 21st century, the number of reported cases to the CDC has grown quite dramatically. It's known that this 30,000 more or less cases per year in the United States is a dramatic undercount of the actual number of cases. But even taking these numbers at face value, Lyme disease is one of the top 3 or 4 most frequently reported notifiable diseases in the United States. And it's by far the most frequently reported vector-borne disease. So diseases transmitted by arthropods like mosquitoes and ticks, fleas, and other arthropod creatures. This, as I said, these reported cases have been known to be a dramatic underreporting of the actual number of cases, and the CDC has concerned themselves with trying to get a better estimate of how many real cases there are in the US. And they've used various methods, including insurance claims and the like, and they just published a paper last week in the journal Emerging Infectious Diseases. And what they now estimate is that prior to 2005, their new estimate is on an annual basis for the previous 5 years or so, 2000 to 2005, there were well over 300,000 cases of Lyme disease in the United States per year. And then in the 10 years following 2005, so 2005 to 2015, there have been almost 500,000 cases per year in the United States alone. So these are enormous numbers, and it cements Lyme disease as one of the top reportable diseases in the US. And for your edification, I can let you know that currently Lyme disease is number 4 in notifiable diseases behind chlamydia, gonorrhea, and syphilis. So I want to mention, even though this talk is largely restricted to Lyme disease, is that there are 2 other emerging diseases transmitted by the same tick that I will describe in a lot of detail in a couple of minutes. So these are very different pathogens, but they're transmitted by the same tick, anaplasmosis in the middle and babesiosis to the right, that although the total number of cases is considerably lower than is the case for Lyme, the trends are alarmingly upward in case reporting in the United States and in various portions of the United States. So as Jim mentioned, the distribution of Lyme disease is somewhat restricted to certain focal areas. This shows the data from the CDC back at the turn of the 21st century, demonstrating that Lyme was largely found in a band along the eastern seaboard from essentially northern Virginia up to southern Maine. A second epicenter or endemic area was the upper Midwest, Wisconsin, and Minnesota. And then I think most of you folks are in and around the Bay Area, so I will point out that California has had a scattering of cases for quite a number of years. But fix this pattern in your mind for a moment, your mind's eye, the upper Midwest, the Northeast, and I'm going to jump ahead a couple of almost 2 decades to 2017, which demonstrates the vast expansion of the area in which you see endemic Lyme disease. It has spread mostly to the north and west from the eastern focal area, and it has spread in all directions from the Midwestern focal area. In California, there still remain some cases that are mostly in and around the Bay Area. And most of what I'm going to be talking about is the ecology of Lyme disease in the eastern half of the United States. In the far west, the players are somewhat different, and there are parallel creatures involved, but it is the western black-legged tick that is the culprit, transmitting the same species of bacterium that I'll describe in just a second. So suffice to say that in terms of number of cases and the geographic distribution of cases, we have an enormous and growing problem, and that's where ecology comes in. So I happen to know that you saw this slide 2 weeks ago, or at least a portion of it, that follows up on some of what Jim said in terms of why we should think that ecology plays a role in infectious diseases, especially human infectious diseases, where the biomedical sciences, which are what we generally think of as the scientific disciplines that are involved in infectious diseases, they concern themselves with things like diagnosis and treatment. And really, ecology does not play a huge role in either diagnosis or treatment of the infectious diseases that plague you and me and our brethren, other humans. And where ecology can play a role, and I agree with Jim that it plays a fundamental role, is here in the third arm, which is prevention. And I guess third arm is a bit of a pun there, unintended. When we think about prevention, we think about a rather non-ecological aspect of it, which is immunization. And we're thinking about that quite a bit when it comes to COVID-19, I think. But prevention, so there is no vaccine for Lyme disease or any of those other tick-borne diseases or most of the mosquito-borne diseases, or in fact, most zoonotic diseases lack a vaccine. So that aspect of prevention is not available to us. And I would argue that that's where ecology comes into play, that these diseases come from nature, especially the zoonotic diseases, and ecology tells us a lot about where those pathogens come from that can make us sick. And that knowledge of where they come from is a fundamentally important aid in prevention, in avoidance of exposure, in making it the case that diagnosis and treatment are unimportant because we can actually avoid cases based on our knowledge of ecological dynamics that go on in and around us. So in the case of Lyme disease, here is a patient showing the characteristic bullseye rash, this characteristic early symptom of Lyme disease. And if a patient walks into a clinic or a doctor's office and looks like that, and it happens to be the right time of year and the right part of the country, then the doctor has no problem diagnosing that as a case of Lyme disease and knowing how to treat it. That would be an early manifestation, and at that point, it's quite treatable. And so this bullseye rash is a nicely diagnostic feature, and it would lead to treatment with an oral course of doxycycline, typically an antibiotic that kills the bacterium that I'll tell you more about in just a minute. But there are many, many cases where a patient does not show a bullseye rash or any rash at all. There are other very generalized early symptoms of Lyme that include things like a fever, chills, muscle aches, lethargy. You feel like a truck ran over you. And so one might imagine that if you walk into a doctor with those generalized kinds of symptoms, and there's no rash and no knowledge of a tick bite, it might be exceedingly difficult to diagnose the patient with Lyme disease. And our blood tests are inefficient and not terribly accurate. So in that case, diagnosis becomes problematic, and treatment of later stage Lyme disease is quite problematic as well. So some people remain sick for very long periods of time. And we have argued that a knowledge of the exposure of Lyme and how to avoid exposure, if we possibly can, would aid tremendously in our public health fight against this tick-borne disease. So here's where ecology comes in. So that patient who walks in with a bullseye rash or without one, with Lyme disease manifested by other symptoms, got exposed to this bacterial pathogen. This is a photomicrograph of a microscopic bacterium. It looks like a worm, but it's not. Those things are invisible. They're so small. That's a spirochete bacterium that's called Borrelia burgdorferi. That's what's causing this bullseye rash. That's what's causing these other symptoms. The only way those bacteria have of getting into a human body or any other animal body is through the bite of a tick. That's the only game in town for those bacteria to get to move around between animal hosts, including humans. And this is the black-legged tick, Ixodes scapularis, that is the culprit in most of North America. And as I mentioned, it has a close relative that is the culprit in the far western United States, the western black-legged tick. But these ticks are born without an infection. Every generation of ticks is hatched out of eggs free of infection with those Lyme disease spirochetes. So they have to get it from somewhere in order to be dangerous and capable of transmitting it to us. And they also bite a lot of different species of vertebrates off in the woods and fields, including mammals, birds, and reptiles as well, especially out there on the West Coast, but also in the southeastern United States. So they have to get it somewhere. They get it from some vertebrate host. And so the interaction between the bacterium, the tick, and one or more of these vertebrate hosts that are ranging out in the wild becomes fundamentally important in determining how high or low is the risk to you and me of getting exposed to this disease. Those interactions in turn play out in exceedingly complex natural systems that include natural undisturbed forest, that include lower density human housing, that include higher density human housing, strip malls, agricultural fields, and the like. So there's an enormous degree of complexity in the natural system, the ecological system, that can influence our probability of exposure to a tick that's infected with the causative agent, this bacterium, and hence our likelihood of getting sick. I would argue that one of the major challenges for ecologists in determining the role of ecology as a discipline in human infectious disease is in understanding the degree to which, how much of this complexity we need to understand. It's an enormous challenge to understand highly complex ecological systems. To what degree can we simplify these systems, pick certain species, pick certain interactions to focus on to explain our risk of exposure? And ecologists sometimes make mistakes. They often do so by oversimplifying. In the case of Lyme disease, there's been a mistake that ecologists have made by focusing oversimplistically on white-tailed deer as the only really important host, which has been demonstrated after about 20, 25 years of intensive study to be an inaccurate appraisal of this system. So we have struggled with this, and I think you'll see how we've struggled with it and how we've resolved our struggles in the next series of slides. So in order to understand the importance of that complexity and to determine which aspects of it we need to focus on, we need to understand the life cycle of the black-legged tick. And that's what I'm showing here in this trace, where what we're looking at is the abundance of each stage in the life cycle of the tick. So let me walk you through this. So eggs hatch in the summer into the first stage, the baby ticks. These are the larval stage. These larvae emerge from the eggs in typically around August, maybe early September in most of the Lyme endemic zone, and they look for a host. They don't crawl very far, maybe a meter or two is the farthest they're able to move on their own. So they wait for a host to come by near them. They are very generalized. They are not picky. They will feed on almost any warm-blooded creature that happens to come by close enough for them to grab a hold, hitch a ride. So if the larval tick happens to find, attach to, grab a hold of a host, an animal host, it will feed for about 3 or 4 days. and during that time, it expands from a tiny little pinprick of a size barely visible into a fully engorged larval tick. It takes 3 or 4 days, one blood meal during the larval stage. After it feeds for 3 or 4 days, it then drops off the host and it drops to the forest floor. And then it undergoes a diapause. It starts digesting that blood meal under the leaf litter or in small pores in the soil. It molts into a nymph and does pretty much nothing for a period of about 10 months. So almost a full year, it is invisible, inactive, basically in a state of suspended animation on the forest floor. Now it's molted into a nymph, so that nymphal tick then activates in the late spring or early summer of the following year. And if this nymph fed as a larva last year on an infected host, it may have acquired that infection, in which case it now activates as an infected nymph that's capable of transmitting that bacterial infection to whatever host it feeds on in this nymphal stage. So these nymphs are similar to the larvae in that they don't move very far. They have to wait for a host to approach them. They are not at all picky about what they'll feed on. And this is their first and most important opportunity to transmit the bacterial infection to a wildlife host or to you or me. Again, the nymph feeds for 3, 4, maybe 5 days, one blood meal in the nymphal stage, and then it drops off and it spends a period of time quiescent, once again, under the leaf litter or in the soil, before emerging late that fall as an adult. It digests the blood meal, uses the blood to fuel the molt into the adult stage, and those adults then take their final blood meal on some medium-sized or larger mammal. They mate while on that host. The female drops off, lays eggs, and dies. And then those eggs, now we're moving back to the left, they then hatch out again the next spring into the next year's larval cohort, which once again is uninfected. So it's a complex life cycle. It takes place over 2 years, and it can involve multiple different vertebrate hosts for the ticks at different stages. One thing we know from various epidemiologic data is that it is the nymphal stage that is responsible for the vast majority of Lyme disease cases in people. So nymphs and adults are both able to transmit, but the nymphs are much smaller. They're much harder to detect. They're active in the late spring and early summer when people are most active outdoors. And we see most cases of Lyme, over 90%, the onset of symptoms coincide with the nymphal peak. So nymphs are really the culprit, and much more so than adults, which only transmit a small number of cases. So what we're really interested in then as ecologists is in trying to understand the 2 main risk factors that determine human probability of exposure to Lyme disease. One is, how many nymphs are there? What's the population size of nymphs? And the other is, how many of those nymphs are infected with the Lyme disease bacterium? Those are the dangerous ones. The uninfected ones will not make us sick. They're harmless. So to figure that out, we go back to this complexity that each of these life stages of the tick feeds on up to dozens of different host species. So if we want to know how many nymphs there are and how many are infected, can we simplify this very complex system at all? And the answer is yes, we can. We can simplify by focusing on this larval blood meal. Because it's the nymphs that are transmitting, how many nymphs and how many are infected are direct consequences of who the larvae fed on. Which hosts allowed them to survive to the nymphal stage, that would influence nymph numbers, and which hosts infected them with the Lyme disease bacterium, which would turn them into potentially harmful nymphs as opposed to uninfected and harmless nymphs. So I'm now going to describe how we figure out the answer to this question, which is, which hosts infect ticks with the Lyme disease bacterium? Which are the pathogen amplifiers is the parlance that we use to address this question. So what do we do to answer that question? Well, this is where we do wildlife biology. So what we have done for a number of years is to go out and set every sized trap you can imagine, live traps, everything from little small traps for the size of creatures the size of a mouse to much bigger traps that catch all kinds of medium-sized mammals. We use mist nets to catch birds. We're trying to catch every ground-dwelling mammal or bird we can possibly catch. And this is work done in and around the Cary Institute in the Hudson Valley of New York. We then return whatever we've caught, hopefully everything that's out there, to the lab. And we house these animals in cages that have a wire mesh bottom. And you can see we treat them really well. So this is a woodchuck, and it has this lovely salad bar. It has a box to hide in so it doesn't get too anxious. This is in the lower left is a southern flying squirrel. In the upper right is a long-tailed weasel. And in the lower right is a raccoon. So we house them, we catch them in August. So that's the time of year when larval ticks are active. And we hold them in these cages with a wire mesh bottom for about 4 or 5 days. And that is the length of time it takes any ticks that are on them, that naturally infest them in nature, to finish feeding in their one larval blood meal on a host, and they drop off. And beneath the wire mesh floors of each of these cages is a pan that catches any ticks that drop off the host. So this is what we do. At the end of our holding period for these animals, we then release them at their point of capture. So none of these animals are euthanized. They're all taken back to where we caught them after about a week or slightly less than a week. So just continuing with this, what we do is, so we collect these replete or these engorged larvae, and we know what host they fed on. We know not only what species of host they fed on, and remember, these are ticks that infested these animals in nature. We're simply bringing the host back to the lab to collect them when they finished feeding. We know the species identity. We also know the individual identity of each of these hosts. You can see, I hope, with this raccoon that there's a numbered metal ear tag on each host that we catch. So we collect these larvae. It is from a known species and known individual of host. We hold them in vials in the lab for about a month. That's how long it takes them to digest the blood meal. And then they molt into the nymphal stage. And then we crush those nymphs, which gives us great pleasure. And we extract DNA. And we conduct PCR, polymerase chain reaction, which is the method that's used basically for the nucleic acid method to determine whether people are infected with SARS coronavirus 2 that causes COVID. So it's a common molecular technique to detect microbes in samples of various kinds. These happen to be tick samples. We test them for 3 tick-borne pathogens, but what I'm going to focus on is the Lyme disease bacterium in this case. So here are the data that are all broken down into one slide. It took us lots of blood, sweat, and tears over several years to collect these data. What I'm showing here is what we call the reservoir competence of each of these host species for the Lyme disease bacterium. And the way to read this slide is that about 90, a little over 90%, 92% of those larval ticks that we collected from a wild, free-ranging, white-footed mouse molt into an infected nymph. So that's a nymph dangerous to people, capable of transmitting an infection when they bite, when they take their blood meal as a nymph. 92% in the case of mice. Trying to find my cursor here. So in the case of the chipmunk, the other small rodent, and then 2 species of shrews, which are small mammals, not rodents, but of similar size. They are infecting between about 40 and 50 or 55% of the larval ticks that feed on them. So white-footed mice are by far the most competent reservoir. They're the biggest amplifier, but these other small mammals are also capable of amplifying to a somewhat lesser degree. In contrast, most of these other hosts are really terrible at transmitting these pathogens to feeding ticks. So gray squirrels, this stands for ground-dwelling birds, not god birds. Skunks, opossums, raccoons, deer, they're all infecting a very, very small fraction of the ticks that feed on them, even if they have in fact been exposed by prior tick bites that they've experienced in nature. So our next question is, which hosts permit the tick to survive while attempting to feed? Now, these vertebrate animals, they groom, they protect themselves against ectoparasites. And they are capable of killing those ectoparasites that are trying to parasitize them. So we wanted to know which hosts are good at allowing tick survival and which are poor at allowing tick survival, because that would help us understand what regulates the abundance of ticks. So we use similar methods for this. They're slightly more involved, a little more invasive. We went out once again and we focused on 6 species that represent different taxonomic groups, different body sizes and life histories of mammals and birds. Again, capturing them in August. This is the time of year when larval ticks are active. Our 6 species were white-footed mice, eastern chipmunks, veeries, that's a ground-dwelling thrush, that's what this is right here, a veery, gray squirrels, opossums, and gray catbirds. Once again, we put them back in the lab in cages with a wire mesh floor, and we held them for 3 or 4 days until all the ticks that were on them naturally had dropped off. They finish feeding, they drop off. Now all these host animals are tick-free, they're clean. We then inoculate them, each of them with 100 larval ticks that we collected locally. And that's what I'm showing us doing with this veery right here. We use this paintbrush, we put 100 larval ticks on these animals, and then we put them back in their cage and we follow the fate of those 100 larval ticks. And I'm dividing their fates into 2 categories. One is that the ticks survive and we collect them during the next 5 days that the animals are held in the lab. The other is that the ticks succumb to grooming or an immune response by the host and are actually killed in the process. So what do we find? Well, again, there's a lot of variation among these different host species in the probability that larval ticks will survive. So this far left bar, our data show that about 50% of the larval ticks that we placed on a mouse survive that experience, and we collect it on the other side as a living, live, engorged larval tick, partially or fully engorged. So mice are by far the most permissive. The 2 birds and the 2 other rodents didn't differ from one another statistically. They were all middling in terms of host permissiveness, supporting about 20%, 20 to maybe 30% or so survival of those larval ticks. And on the far right, opossums were groomers extraordinaire. They were terrible hosts. Even though ticks feed on them in nature, the opossums were killing about 94% of, 96%, as I recall, of the larval ticks that were trying to feed on them. So this variation among hosts matters a lot. The fact that white-footed mice are both permissive, they allow the ticks to feed, and this is a close-up of a typical mouse here in Millbrook, New York in August of the year. All those little shiny blobs on the ear of the mouse are larval ticks. they are really by far the most permissive host for tick feeding and the most likely to transmit an infection to those feeding larval ticks. So what that means is that if there are lots of mice running around in the woods, then each new cohort of larval ticks that, again, hatches free of infection has a high likelihood of encountering a mouse as opposed to some other host. That means that many of those larval ticks will survive that experience, and many of those larval ticks will acquire an infection from that experience. And as a consequence, at a later date, because of the life cycle of the tick, the delay, there will be lots of infected nymphs and highly elevated risk to you and me if we happen to walk out into tick habitat, which is almost any terrestrial habitat. So lots of mice means lots of infected ticks. So what I want to do, I think I have our pause right there. And so I guess we can take a break right now, but I wanted to give an opportunity, since I focused so much on these data specific to Lyme and hosts and ticks, tick life cycle, to see if there were any questions. I'm not entirely sure how to do the questions, but I guess people can unmute themselves and ask, or Jim, should we do questions now? Should we take a break and then do questions? How should we do this?
I think
This
what we
pause.
can do is we can take a break. People can do whatever they need in the next 5 minutes. Let's take about a 5-minute break. That's probably all we need in the interest of time management. And then any people that might have, you want some questions answered or discussions during that time, I'll keep an eye on the time and then we'll start again in about 5 minutes. So let's do it that way, okay?
Sounds great.
Okay. So you guys, those of you that want to chime in at this point can unmute, please, and go ahead and do that. And if I'm going to give everyone else an opportunity, and then if people don't have questions, I'm going to ask one. So take your first shot if you like, and then if not, I'll jump in here. Okay.
Can I ask what the technical problems are that make testing for the disease problematic?
Yeah. So the same test is used now that has been used for about 25 years, and it's a serologic test. So you have a blood sample drawn, and what the lab test looks for is antibodies to Borrelia burgdorferi, the Lyme disease bacterium. And it's a 2-tiered test. I won't go into the details, but both tiers of the test have a fairly high false positive and false negative rate. They're combined in such a way that the combined false positive and false negative rate goes down, but it's still actually unacceptably high, in my opinion. So there can be cross reactivity. So you can show an antibody response that is not specific to the Lyme disease bacterium. That would give you a false positive. You're showing antibodies, but it's not necessarily to that specific pathogen. On the other hand, sometimes you can have a low titer of those antibodies, and you are producing them, but they're not at detectably high levels, and that would give you a false negative. So typically what people would do at that point is to focus on detecting the DNA of the bacterium. That becomes problematic because Borrelia burgdorferi, the Lyme bacterium, leaves the bloodstream fairly quickly after infection, and it moves into other tissues, a bunch of different other tissues. And so you can take a blood sample and fail to find Borrelia, even though the person's synovial fluids or cerebrospinal fluid or connective tissue or heart or bladder are infected. And so it's been problematic to come up with a good test either based on antibody production or direct detection of the pathogen and its nucleic acids. There are a lot of labs working on this, but progress has been quite slow, and the Lyme patient community is very frustrated with this, needless to say.
Thank you.
I see Cindy's hand up, yes.
As a person who lives out in the redwoods, I get tick bites regularly, and mostly from my dog. I think they jump off of her, and I get them right away. I mean, I feel them bite, and my husband is very good at picking them out. And my doctor says if I get them out within 12 hours, don't worry about it. They're not engorged. Don't bother with any of these silly tests. Just go on with your life. It hurts. It swells a bit, and that's all. Is there something about how long the tick is in you?
Yes, yeah, there is, and it takes these Borrelia a while to get from the tick into your body, about a day. So if you do detect and remove the tick within 12 hours after it attaches, then you're in good shape. Sometimes you don't really know when it started to feed, so your guess as to whether you're within that day-long window might not be so accurate. But the bottom line is that the sooner the better to prevent transmission.
Thank you.
A little nerdy stuff for those who are interested. The reason it takes so long is that the Borrelia bacteria hang around in the digestive tract of the tick. It's the midgut of the tick. That's where they live. That's where they're hanging around. And then when the tick starts to drink blood, so it's embedded its mouth parts, it's starting to suck your blood in vampire fashion. Some blood enters the midgut, starts to flow into the midgut super slowly. The Borrelia detect the chemical signal from the blood entering the midgut. And what they do is they tear a hole in the lining of the gut, and they tunnel through the gut and to get into the circulatory system of the tick, which is called the hemocoel, which is the space surrounding the gut. From there, they swim upstream to get into the salivary glands, and because that is their pathway to get out of the tick through the mouth parts and into the host. And all that takes them at least a day, even up to 2 days to do. They do this all very slowly. So that's lucky for us that we have a little bit of a window, but the sooner you can pull the tick off, the more likely you are to prevent that from following through the whole way to infection. Enough nerdy stuff. We're going to get a lot more nerdy stuff going forward in the talk.
Okay, Rick, I think we're now at my iPhone says 10:49, which is just about a little over a 5-minute break. So why don't we end this and go ahead and finish up the remainder of your lecture, and then we can finish up with discussions when you're done with that. Okay.
Okay, sounds great. Okay, so this is where we stopped. We're back to screen sharing. You can all see lots of mice equals lots of infected ticks. We're good with the screen sharing still?
Yep.
Yes, okay. All right. So I've presented evidence to you that lots of mice means that larval ticks will have excellent opportunity to feed on this particular creature, which is excellent at promoting tick survival and tick infection. So then the question becomes, what produces lots of mice? Can we predict it? So if we can, then we will be able to predict when Lyme disease risk will be elevated. And so one of the main answers to the question, what produces lots of mice, turns out to be lots of acorns. So this is true of all kinds of different rodents, forest-dwelling rodents, where episodic production of large seeds by the dominant forest trees can cause these consumers, these mouse consumers, to go through rapid population growth. And the reason for that is that these acorns and other large forest tree seeds are an excellent source of nutrition. The acorn weighs a couple of grams, the mouse weighs about 20 grams, so it's really big. It packs a lot of lipid, protein. There are certain types of acorns that have a very long shelf life. They're protected by tannins from decomposition, so they can be stored. They can be gathered in large caches. And so when there are lots of acorns around, the mice store them in very large quantities, and they use them as an overwinter food supply because they're produced in the autumn, they're collected in the autumn and winter. And so the mice are able to remain in good body condition during a time of year when food is generally poorly available. They even are able to breed sometimes in what's typically a non-breeding season. If there are lots of acorns around, they get a jump start on breeding the next year, and they can undergo very rapid population growth. So at our sites at the Cary Institute that we've been studying these phenomena at for a few decades now, we find documentation of this hugely variable acorn production from one year to the next. So these are the acorns produced per square meter of forest floor between 1992 and 2018. So I want you to note that there are some years when there are no acorns produced at all, or very few produced, such as in 1999, in 2004, and again in 2007, etc. And other years where there are copious quantities of acorns produced throughout the forest. And I want to point out that our years of the highest peaks in acorn production at the Cary Institute were 1994, 1998, 4 years later, then 8 years after that in 2006, 4 years after that in 2010, and 5 years after that in 2015. So we have been monitoring the population size of small rodents, including white-footed mice, over that entire time as well through a live trapping capture, mark, release program. And here are the data on mouse population size by grid over that time. So that first acorn year, if you recall, and I'll go back if you don't believe me, was 1994. 1995 was this big peak in mouse population. Then again, 4 years later, we saw a big peak in mice 1 year following the next acorn peak. Then 8 years later in 2007, 4 years after that in 2011, and then 5 years after that in 2016, where each of those peaks in mouse population is one summer after the heavy acorn year. So part of the answer to the, what makes for lots and lots of mice is lots and lots of acorns the previous year. And we've used these data to make the prediction that if acorns in 1 year result in lots and lots of mice the next year, and because of the life cycle of the tick, we've argued that lots of mice in that next year should result in a lot of larval ticks being able to feed on mice as opposed to some other host. That should lead to lots of infected nymphs 2 years after the acorn year, or 1 year after the mouse peak. And we've analyzed those data from the Cary Institute and found that indeed there is a significant positive relationship between the production of acorns 2 years ago, that's what this T-2 year means, and the abundance of infected nymphs in the current year. So it's a significant predictor of our risk of exposure to Lyme disease. It's a leading indicator that is present almost 2 years in advance. So 2 points that I want to make from this. One is that we have used acorn production and mouse population size as a predictor in a way to try to foster prevention and avoidance of Lyme disease. We have made ourselves available to journalists of various kinds, to public health experts, and we've publicized when we think Lyme disease risk will be elevated, and we do so in advance so that the protective measures that people can take are the topic of discussion, education, training, and how to protect yourself against Lyme disease. The other thing that I want to mention scientifically is that this is a positive correlation, and it's a statistically significant one, but there's a lot of variability in the data. There's a lot of scatter. And so what that means is that it's not only acorns that are influencing how many infected nymphs there are in any given year. There are other factors that are not present in this analysis. And we study those too. There are things like weather and climate, and those are topics of our work that I don't have time to talk about. But I do want to indicate that acorn production and mouse abundance are not the only factors that influence how many infected nymphs there are, but they are big ones. They produce a large signal. So I want to summarize that, because this is a complex series of relationships between creatures over 2 years. So what I've tried to argue is that the size of the acorn crop 2 years ago is a major determinant of the size of the mouse population last year, and that in turn is a major determinant of the size of the infected tick population this year. That is the risk factor that influences our exposure to Lyme. I also want to point out, with this big blue arrow coming down from above, that there are other important factors under study that influence the mouse population and hosts more generally, and that's what I want to turn to next. In essence, in ecology, when we're interested in the population of a species like the mouse, or you choose an animal, some kind of consumer, there's sort of 2 paradigms. One is what we call bottom-up forcing, where, and the bottom-up is a metaphor for a food web in which we array the resources at the bottom, the consumers at different trophic levels above them vertically in a conceptual model. And so bottom-up effects would mean that the food supply, in this case acorns, is influencing the size of the consumer population. But then there are things that eat the mice, there are things that compete with the mice, there are things that infect or infest the mice, and those would be considered top-down forces that might regulate the size of that population. And we know those exist as well, and I'm going to describe a few of those phenomena next in the context of the ecology of Lyme disease. So another component of our field work has been to understand the role of habitat fragmentation in influencing our probability of getting exposed to Lyme. What we've done is gone out to a series of different forested sites, dozens of them in New York, Connecticut, and New Jersey. And we have done intensive biodiversity surveys using traps of various kinds, camera traps, mist nets, and the like, to get a handle on what species occur in those forested sites, what vertebrate species occur there. And we've arrayed our sites from large expanses of undisturbed forest to somewhat smaller fragments of those forests to really small fragments of those forests on the order of an acre or so, acre or two in size, even smaller than an acre. And what we've found is that as we chop up large expansive forest into little bits, little patches, we lose species. A bunch of the species are no longer detectable there and appear not to occur there at all or are very scarce. But interestingly, we're not just losing species at random. And this is a pattern that has been demonstrated with other habitat types and other groups of species. You tend to lose the larger bodied species, the more ecologically specialized species, so the ones that rely on only a few food sources or a few habitat types or nest sites, and the ones that are more carnivorous. They tend to be the ones that disappear when the size of their habitat is reduced, as is happening here on this array of forest fragmentation. So what you tend to lose are the predators, the large-bodied animals, and some of the forest specialists that need a larger forested area for a viable population to occur. And what you tend to have remaining are the small-bodied, more omnivorous, generalist species like the small rodents. And recall that the small rodents are really important culprits in producing tick populations and infected ticks as well. So we predicted, based on these biodiversity surveys and the species that disappear when you chop up the woods into little bits, that these small forest fragments would be the places in which we would find the highest numbers of infected ticks, especially of the nymphal stage, as I've been saying, is the most dangerous stage. And indeed, this is exactly what we've found in a paper published quite a while ago now. What we found is that this is the density of infected nymphs, the population size of infected nymph stage ticks was considerably elevated in these small forest patches. These are areas that are about 2 acres or so in size, 1 hectare more or less. And in somewhat larger forest patches of 3 to over 7 hectares in size, we see much reduced abundances of infected nymphs. Interestingly, one of the things that happens is we carve up the forest into small bits. We are elevating ecological risk because we're producing large populations of infected nymphs in those forest patches. And then many of the developers are carving up the forest in order to install housing developments. So we're placing people's backyards right up against those newly highly risky habitats that we've created by fragmenting the forest, inadvertently, of course. But it's an important dynamic that needs to be corrected. So habitat fragmentation selects for certain species like mice and selects against other species. And I want to talk about that a little bit more, to talk about work that was led by a former UCSC graduate student, PhD student named Tal Levy, working with me a few years ago as a postdoc. And he set up camera traps in sites throughout the county that I live and work in, Dutchess County, New York, to ask about the relationship between the guild, the collection of predator species, and how many of the ticks are infected with Lyme disease bacteria. And what Tal and the rest of us found is that sites that have a full diversity of predators have about 50% reduced tick infection. So sites that have, especially things like this red fox that was captured in this camera trap, tend to have much reduced tick infection. So in essence, what we have found is that more diverse animal communities have more host species that do one or more of these three things. They either attract ticks and kill them. Remember our friends, the opossums. We actually consider opossums as ecological traps, that the ticks will climb onto an opossum at their peril, and they tend to get killed when they do so. Other hosts fail to infect those ticks that get on them and do survive. So remember, almost every host other than white-footed mice was a poorer reservoir for infecting the ticks. And then this work by Tal Levy and the rest of us showed that some of these animals in these more diverse communities, especially foxes and to some degree bobcats, are able to regulate the abundance of rodents, which therefore reduces the probability of tick survival and tick infection. So when we noticed this relationship in the Lyme disease system, and by we right now, I mean Felicia Keesing and me, we are, she's your first lecturer 2 weeks ago in this system. She's my collaborator in many things, both science and personally, because she's also my wife. We coined the term dilution effect to characterize the situation in which high diversity leads to low disease transmission. Or conversely, if diversity is reduced, that should elevate disease transmission. So we call this the dilution effect, and there's been considerable interest in whether this relationship between diversity and disease pertains to Lyme only, or whether it's a more general phenomenon across various different kinds of infectious diseases. So I'm going to turn now to work that was done by others, not in my lab, and now we're broadening this conversation to extend from the Lyme disease system. And back in 2015, this paper appeared in the Proceedings of the National Academy of Sciences, one of the world's leading scientific journals. Work done by a group from the University of South Florida. And what these researchers did was they conducted a meta-analysis. And a meta-analysis, I'm going to try to move this thing. A meta-analysis is an analysis of analyses. So in order to do a meta-analysis properly, one needs to do a complete literature search. So you can't just pick and choose the scientific papers you want to incorporate in your meta-analysis. You have to do a comprehensive literature search. So that's what these folks did. And what they did was they found a literature that consisted of 202 different studies, and that's what they consider effect sizes, that are on 61 parasite species. So they're using the term parasite broadly. These are parasites and pathogens. This includes viruses, bacteria, worms, etc. So this is a comprehensive analysis of the studies that look at the relationship between diversity and disease. And they calculate a statistic, which is called the Hedges G statistic, that assesses that the nature and strength of that relationship between diversity and disease. And it is not advancing for me. There we go. So this first component of this multi-panel figure shows for all parasites, so across all 202 different studies, 61 different parasites and pathogens, what is the relationship between diversity and disease risk where they're measuring disease risk by parasite abundance. What's the relationship between diversity and parasite abundance, animal diversity and parasite abundance? What they find is that it is very strongly negative. This is at about minus 1.1 or so. So what that means is that the higher the diversity, the lower the parasite abundance. It's a negative relationship. And this star means that it's statistically significant. Generally, when you get a Hedges G value that is below minus 0.5 or above positive 0.5, that's considered a strong effect. So this panel A here shows that across the board, across all these different studies, there is a strong and consistent negative relationship between diversity and parasite abundance, where more diverse systems have fewer parasites. It turns out there are more interesting aspects. I won't go through every one of these panels, but I think they're quite illustrative. It turns out in this panel B that it doesn't matter whether the parasite attacks wildlife species or humans. It's significantly negative in both cases and not different between the two. It doesn't matter whether the parasite has a complex life cycle, like some worms that might go through 2 or 3 or 4 species of host before they reproduce, or a simple life cycle, like a bacterium or a virus that goes from one host to another. It doesn't matter whether the parasite is a bacterium or a virus, which are the microparasites, or whether it's a protozoan or a worm, which are the 2 of the macroparasite types. And then skipping over to panel F, it doesn't matter whether the study being evaluated manipulated diversity in the field or in the lab as an experimental study, or it did not manipulate diversity, but simply observed naturally occurring diversity and compared them, which would make it an observational study. And then in a slightly more recent paper by Wang et al. From a couple of years ago, they did a virtually identical type of analysis, but now they're comparing diseases of animals to diseases of plants. And once again, they find a strong negative relationship. And once again, they find there's no difference between diseases of animals and diseases of plants in terms of this negative relationship between diversity and the abundance of the parasites causing disease. So, even more recently, there's a group that got a little more into trying to understand the mechanism by which this relationship exists. So this is a paper that was published in Nature back in July of this year. And this group used publicly available data sets all over the globe to ask about how host animals and non-host animals change in their diversity and abundance across gradients of disturbance by humans. Okay. I'm going back here. Okay, so this map shows you the large number of sites for which this group, this is out of the UK, the large number of sites for which these folks had data on the animal species that occur across gradients. So at every one of these sites, they sampled biodiversity of various animal groups across a gradient from relatively pristine, undisturbed habitat to moderately disturbed or recently disturbed habitat to heavily managed habitat like crop fields and other agricultural systems to heavily disturbed areas represented by urban areas. They asked which animal species occur across those gradients and how many of those species are hosts for zoonotic pathogens that can make people sick. Okay, so that's the nature of this global scale study that Gibb and colleagues undertook in this study published in Nature last year. And this is their most important results figure from the paper. It's a little complicated, so let me walk you through this. What we're looking at here is the gradient. So in this panel A, we're going from primary, which is the undisturbed natural habitat, into secondary, which maybe have previously disturbed and it has recovered somehow, to the managed, this is the agricultural or suburban land, and all the way to the urban on the right. Okay, so what the data show, these kind of, on my screen, they're sort of turquoise symbols. What they're showing is these are the non-host animals. This is the difference plus or minus from the primary habitat. So in essence, what's happening is that non-host animals, as you go from somewhat disturbed to more heavily disturbed to very heavily disturbed areas, they're the ones that are declining or disappearing. Some of the species are disappearing, and they have other data showing that the ones that remain are much more scarce. So as you go across this gradient from undisturbed to heavily disturbed land types, you are losing the non-host animals. Conversely, these brown symbols here show that as you go across that same gradient, they are thriving. You have more species of host animals in the more disturbed areas, and other figures, other data in this paper show that you have higher abundances of those animals as well. So in essence, what we are doing inadvertently when we disturb habitat along this gradient is we are imposing a filter on what animals get through and occur in our most disturbed, most human-dominated areas like these urban areas. And the filter weeds out the non-hosts. They don't make it through by and large. But the species that do make it through are the ones that are dangerous, capable of making us sick, because they are the hosts to zoonotic pathogens. So Felicia and I were invited to write a commentary on that paper in the same issue of Nature that came out last summer. And so just reading their byline, the journal provided this byline, we didn't, but it asks a question. It says, does the conversion of natural habitats to human use favor the proliferation of animals that harbor agents causing human disease? And the answer to that question, based on the Gibb and colleagues paper, is yes, most resoundingly yes. So this appears to occur at a global scale. So I want to sum up here and then leave, I hope, ample time for discussion afterwards. Returning to the Lyme disease system as an exemplar system, Lyme disease risk is elevated when mouse abundance increases, and that predictably occurs following acorn years by this bottom-up forcing that I was talking about. Risk is elevated when biodiversity declines, which turns mice into the dominant species occupying those low diversity habitats. And that occurs when forests are fragmented through the loss of predators, probably competitors, although this is less well studied, and poor hosts that may not act as predators or competitors when they decline. And that is results in this increase in risk of human exposure to Lyme. The Lyme system has generated what I think are general principles that apply across many different infectious diseases. And those are, more generally, that host species, for any given pathogen, host species vary quite a lot in quality for the pathogen. This is essentially always the case. There are some hosts that can amplify the pathogen, and others that simply don't, and yet others that are in between, that amplify it maybe slightly or regulate it slightly. It is also generally the case that the amplifying species are the ones that thrive when habitats are disturbed. That is what we found in the Lyme system. That is what Gibb and colleagues found across the globe in this analysis across those gradients from all over. And then lastly, the impact of these pathogen amplifying species, these so-called reservoirs, can increase when diversity is lost, because some of that diversity is capable of regulating them or the pathogens directly. When resources surge, it doesn't have to be just acorns. There are other resource pulse systems that drive consumers through ups and downs. And also when predators decline, because many of these pathogen amplifiers are found at lower trophic levels. They are not themselves predators, but they can be controlled by predators. And if predators decline or disappear, then that regulation can be weakened and they can be more abundant. So I want to stop there so that we have ample time for questions and following discussion. So maybe what I should do is stop sharing my screen and open the floor. Is that a good time to do this now, Jim? Can we open things up?
Yeah, it's fabulous, Rick. Thank you. And thanks so much for a very clear and informative presentation. I have some questions, but I'm going to defer. I'll ask all of you that might like to chime in here to go ahead and unmute your mic so we can hear what you have to say. And then I'll defer the several comments or questions that I would like to put forth, but open it up for any questions or discussion. I saw there was a question by Pat that, and I believe your question was, do any of the other hosts suffer? And I presume you meant, do other things get as sick as people from Lyme disease, these other species that carry it? Was that the correct question there? That's a great question. And I wondered, Pat, am I representing your question properly if you're still there?
Yes.
Okay. Right. So, so a Borrelia infection, an infection with the Lyme disease spirochete, generally does not have strong pathologic effects on the few other non-human animals that have been studied. Dogs get quite sick when they're exposed. They can get similar symptoms to humans, arthritis, fever, lethargy, and the like. And horses can get quite sick. And these are both phenomena that veterinarians and owners of dogs and horses are quite aware of in Lyme endemic areas. We've looked at whether white-footed mice are affected by an infection or not. And we've looked at that by actually giving them a vaccine. You can vaccinate mice, can't vaccinate people. And we found no detectable effects of clearing an infection in mouse populations on either their behavior or their survival or reproductive rate. There are other wildlife species in which folks have looked for various pathologies. arthritis, lesions, and the like. And these scientists tend not to find much of any effect of Borrelia burgdorferi infection on health of these other wildlife species. So there are only a few, and we don't really understand why dogs, horses, and people get sick, but mice and squirrels and deer, for instance, don't appear to. We don't understand that, and I think it's an interesting aspect of comparative biology and even comparative immunology. But when I speak about this difference at conferences where there are immunologists, like real immunologists, they just kind of laugh at me when I suggest that this is an opportunity to understand why different wildlife species show such different responses. Some completely tolerate infection with Borrelia and don't get sick. Others seem to completely resist it and don't get sick. And we do get sick, but we don't know why we do. But I'm very naive, I guess, in thinking that this is a topic that's worthy of study by comparative immunologists. They don't have reagents to work with opossums and white-footed mice and raccoons. They aren't interested. So there are many mysteries about the impact of this bacterium on wildlife hosts.
Hey, Rick, Dan Costa here. You may have made a comment on this, and I missed it during the break. There's a big body size effect, and so part of that could be that the habitat. And you may is it because mice are rooting around in the leaves in a microhabitat, and a deer is tall and basically is not really coming into contact with underlitter and such, or is it something about just body size intrinsically that dilution effect? One shot into a mouse, which is tiny, is going to be a much greater percentage of blood volume and body mass than the same inoculant into a deer.
Yeah, that's a great question, Dan. I appreciate that one. There is a strong body size effect. If we have plotted the relationship between that reservoir competence, right, the probability that a host will transmit Lyme infection to a feeding tick, and the body size of that host. And it's a very strong negative relationship. So body size is responsible for, or at least correlated with, inversely with the ability of the host to transmit. We don't know why that is. Larger bodied animals do tend to have more ticks on them. So deer have plenty of nymphal ticks on them. Raccoons do, skunks, they all have more than mice, but whether it's it doesn't scale to the mass of the host. So even though in absolute terms there are more ticks on them, per gram of host are not more. But then whether there's, that would be another dilution effect, I guess, that there's more blood volume to dilute the presence of these bacteria. We don't know what it is about large body size that makes these big animals poor reservoir hosts and the small ones really good reservoirs. I can say that I've worked with a couple of eco-immunologists, and there is an interesting relationship between immune strategies and other life history strategies that have been found in the Lyme system and in a few others. And the basic idea is that these small-bodied species have evolved a kind of a live fast and die young lifestyle. So they reach sexual maturity at a very early age, a month or two. They have very large litters, 4, 5, 6 babies. Every month or two, they immediately become reproductive again after giving birth. And then they drop dead from exhaustion because they've been producing so many babies so quickly. And those correlated with that live fast and die young life history is what appears to be a tolerant immune strategy, where they do not expend much energy mounting expensive immune defense. They opt instead for tolerating infection. And it may be the case that their fitness, the fitness of these individuals, is more tied to their ability to produce as many progeny as they possibly can before they get nabbed by some predator, as opposed to trying to protect themselves against infections so that they can live a long time and have that 1 or 2 or 3 babies once a year or every couple of years for an extended period of time. If that's the case, then generally you would expect the small-bodied, ecologically very resilient species to be the ones that tolerate infection and therefore allow proliferation of pathogens in their bodies. And that's what makes them good reservoirs for zoonotic pathogens. So there is some research on that relationship between life history strategy, immune strategy, and function in a zoonotic disease. But not enough, I think, to nail down that relationship or really evaluate it as rigorously as I think it needs to be.
That's really cool. It really, you're right. It fits into the pace of life scenario extremely well. That's really cool. Thanks.
You bet.
So I'm just going to take over as a manager here because there are a bunch of great questions coming in on the chat, and
Okay.
I could just ask those of you who asked those questions, but I'll just go ahead and ask them. There are a couple here, and just one that could probably be answered fairly quickly, Rick, is the question of what is the mortality rate, human mortality rate for Lyme disease? How many people die, or what percentage? What's the likelihood of that?
Right, I just opened the chat. I'm not seeing that one, but it's very low. There are occasional reports of carditis that bullseye rash I showed on that person's, I think it was somebody's leg. There was a mortality here, right down the road from where I live and work in Dutchess County, of a young man, otherwise extremely healthy, who collapsed and died very quickly. And in the autopsy, they found essentially that same bullseye rash, but in his heart. So there are occasional myocarditis manifestations of Lyme that can be fatal. That's far less than 1%. So Lyme is extremely rarely fatal. Those other tick-borne diseases that I described, anaplasmosis and babesiosis, although they're much less common than Lyme, they are slightly more commonly fatal. There is one virus transmitted by that same black-legged tick. It's called Powassan virus, and it was named after a town in Ontario where that virus was first discovered. And that has about a 15% case fatality rate, which is quite high for an infectious disease. And of those people who recover, don't die, I shouldn't say recover, who survive, about half of them have a permanent disability because the virus causes encephalitis. And so they can be disabled long-term or even permanently. So that is exceedingly rare, Powassan viral encephalitis, but it is a tick-borne pathogen that is very concerning. If it were to emerge, it would be quite problematic in our area.
Great, thank you, Rick. Most of the other questions that are posted up here are management related questions. So arbitrarily kind of go through the order in which they were received. And there's a question up here from Pat, I guess, about lizards in the West, and are they a buffer by way of the dilution effect? I think that's probably as simple a way of putting the question as any.
Yeah. So so out west there, these ticks do bite lizards a lot, as well as small mammals and medium-sized mammals and birds and deer. So the Western black-legged tick is similarly a very generalized parasite. And it turns out that there's a researcher at who's now retired at UC Berkeley named Bob Lane, who with colleagues discovered that Western fence lizards have a circulating protein, so it's part of their innate immune system, that kills Borrelia in the tick. So it kills the Lyme bacterium in the tick before the bacteria are able to leave the tick and enter the lizard's bloodstream. And it was thought that that lizards were likely, these Western fence lizards were likely to regulate Lyme disease risk. More lizards means less Lyme disease risk because they're killing the bacteria in the tick. It's a very complicated story, but there is a professor at San Francisco State University who, for her dissertation at UC Berkeley, among other things, did a large-scale replicated lizard removal experiment. So she noosed lizards and took them out of certain areas and then released them into the counterpart areas. So you had low lizard and high lizard areas. And she found that actually when you remove, let me get this right, I was a co-author on this paper. So when you removed lizards, the abundance of infected nymphs went down. So the expectation was that they would go up because you're taking away the species that cleanses the lizards of their infection. And it turns out, boiling a complicated story down somewhat, is that lizards have two distinct and different impacts on the abundance of infected ticks. One is on abundance and the other is on infection. And those work in opposition to each other. So although the lizards, more lizards means less infection because they're cleansing the ticks, more lizards means more absolute numbers of ticks because they're a permissive host. The ticks do really well feeding on lizards. And so the net effect on the abundance of infected ticks is the combination of those two independent effects. And it turns out that lizards elevate abundance of ticks to a greater magnitude than they reduce infection of the ticks. So the net effect was more infected ticks when you reduce the lizard population. So that's probably more than you wanted to hear. The woman's name who did that work is Andrea Sway, S-W-E-I. So if you want more details, you could do a search on her work with Western fence lizards and the Lyme disease system. She's still active, quite active in this field.
Hey, Rick, thank you. I'm going to move to the next question, which again is a management related question, and that has to do with the degree to which burning and fire, especially through management control burns, may have an impact on Lyme.
Yeah, this has been studied to some degree, and it's very controversial. And I think you get different answers. And so people fight with each other about whether prescribed burns are a good management tool or not. My read, I haven't done any of this literature. My read of it is that the controversy exists because of the different intensities and extents of the prescribed burns. So typically in the East, when there's a prescribed burn, you indeed get a reduction in ticks, but it's very short-lived because there's rapid regrowth of vegetation here. This is a more mesic climate here. Burns tend not to be super hot. They tend not to cause massive tree mortality. They more cause mortality of plants in the understory than in the overstory. And so the effect of most prescribed burns is to temporarily reduce habitat for ticks, which rely on shade and moist understory conditions for survival. But that following the regrowth following burns attracts wildlife rampantly, and those wildlife import populations of ticks, and very quickly you have the restoration of more mesic, equitable conditions for the ticks to exist. And so, generally speaking, prescribed burns in the Lyme disease endemic area, the upper Midwest, the Northeast, is thought to be ineffective and maybe even counterproductive. That is different in the Midwest and southern states, mostly where you're dealing with Lone Star ticks, a somewhat different behaving species, also transmits illness, and they're a really, really annoying tick, much more so. They chase you down, they kind of hunt you, unlike the black-legged tick, which is such a wimpy crawler in nature. Lone Star ticks will chase you actively. And prescribed burns are both more intense and typically larger in extent, and have been shown to do a better job for a longer period in reducing tick population. So it's a long-winded way of saying that a lot depends on the type of burn, the location, and the type of tick you're trying to control. But it can be effective under certain situations. It probably would be in the West. There's a little bit of work that's been done in the West Coast by a fellow named McDonald, who was a UC Santa Barbara PhD student. So there is a little bit of work done in California on prescribed burns and ticks, and I think it is fairly strongly inhibitory.
Great, thanks, Rick. The next question here is kind of has to do with the geography of the risk and the spread of risk, and the question of why it's so prevalent in the Midwest and East compared with out West here. And let me just ask that question a little differently, that more fancifully, and to say that it looks like there are really two kind of major epicenters, the Midwest and the Northeast, and then there's a big expansive habitat in between that isn't fundamentally different. It's still eastern deciduous forest habitat. And so what do you attribute those patterns to? Not just the one I specified, but I mean, what is the limiting effect that has prevented some areas from being outbreak areas and other areas not?
Well, I would say one of the answers to that question is time. You know, Ohio and Indiana will be in the Lyme endemic zone within 10 years, I would wager. Both those epicenters in the upper Midwest and in the Northeast are growing and expanding. I think that you know, corn and soybean fields are not good habitat for ticks. So to some extent, if you have those sort of those extensive agricultural fields, you will not get much Lyme disease because the ticks can never get very abundant there. So right now, for reasons that we don't understand, there is dispersal limitation. I think there clearly are areas in North America where the habitat is suitable for the ticks, but they haven't really arrived in numbers. And so we don't have a detectable Lyme disease problem there. They will be invaded, I think, that area between that you're pointing out, Jim, between the Midwest and the Northeast. There is also a fair bit of evidence now to suggest that some places that were either inhospitable to ticks or marginally hospitable to ticks are getting more favorable because of climate change. So there are a number of studies now that show that mountaintops or high elevation areas in the Midwest and Northeast are being increasingly invaded, as are higher latitude areas as well. And that appears to be linked to a longer warm season. So the ticks need a fair bit of time to find hosts 3 times in their lives. They need an extended warm enough period where they can seek hosts before they have to go back down underground. And if they fail to find a host, they die. And if too many of them die, you don't have a population. So it looks like climate change is lengthening the duration that ticks can seek hosts. And by that mechanism, it is opening up habitat that currently or formerly was too harsh for them to invade. But they have to get there too. And this is where we really don't understand what is moving ticks around at large geographic scales. Everyone always says, well, it's birds. And it probably is, but we don't really have much evidence for it. And the birds don't just move around willy-nilly. There are sort of traditional migration routes. They move at particular times of year. Those may or may not correspond to the seasons when the immature stages of the tick infest birds. So it's a little facile to just say, well, birds will move them around. And then it's only a question of whether viable populations can be established based on hosts and abiotic conditions. We know a lot, we know very little about the gross, large geographic scale patterns of tick movement, which has to be on hosts, but we don't know which ones, when and where they're being moved, whether they're being moved by things that also infect them or things that don't, and that matters. So there's a lot of work left to do. This is part of the question about how much of the complexity of these natural systems do we need to focus on to understand what we want to understand, either for management or for basic knowledge. And everyone draws that line in a different place. So these are hard questions to address. And I think that explains why we don't know more right now. There's a lot of interest, but not much funding, frankly. And not that many people working on this in an ecological sense.
Couple more questions and then we'll be right at noon, and I think we'll pull this to a close. I'm going to ask one that I know you can answer because I know it's the centerpiece of a paper that is recently in press that you and Felicia just wrote, and it's essentially how do you determine sort of the balance between the amplification and dilution in having to do with biodiversity? That's a hard
Yes,
question.
another wonderful question. And so in one sense, you know, what I showed you that the meta-analysis by David Civitello and colleagues, and the other one by Zheng Wang, those data represent the net effect. So they are asking what's the net effect of diversity. That if there were a positive relationship between diversity and disease, that would constitute an amplification effect, as Felicia Keesing and I, we collaborated with Bob Holt on that study. That's how we defined an amplification effect. So some of the studies are actually asking what's the net effect. And if it's negative, net effect through multiple mechanistic pathways, the net effect of diversity on disease is to reduce disease, then that's a dilution effect. If the net effect is to increase, that would be an amplification effect. So that's kind of a simple level answer to that question. There's a lot of confusion in this literature. It's somewhat controversial, I think, to some degree, needlessly controversial, but nevertheless, it is. One of the areas of confusion is, what do we mean by disease risk? And some people define disease risk by simply the net number of different pathogens. So if there are more pathogens, things seem riskier, right? There are just lots more things that can make us sick. And given that those pathogens come from animal hosts of various kinds, then it stands to reason that if you have a high diversity of animal hosts, you're going to see more total diversity of pathogens. And isn't that concerning? So doesn't that mean that we're more likely to get sick from something? And so that has led to the thought, the postulate, that it's those really diverse places on Earth that have lots of different animal species that will have lots of different pathogen species. And that's what we need to worry about. So wild nature is dangerous, and we should be concerned with it, or maybe just get rid of it. You know, maybe Trump being an ex-president is a bad idea, because he would have happily gone and paved it all over, or turned it into casinos or high-rise towers. That maybe we should get rid of diversity, because those wild, highly biodiverse places are the risky areas. And that's simply not borne out by the evidence. The main reason it's not borne out by the evidence is that that diversity of pathogens doesn't do what you think it does. In essence, most of the pathogens that can infect humans come from only a small fraction of the animal diversity. It's those creatures I've been talking about. I think Felicia talked about rats, not rhinos, right? So it is only a few of the animal species that are capable of transmitting most of the pathogens. And it's a predictable group of the animal species and a predictable group of the pathogen species that are capable of making us sick. It is the ones that we've been talking, it's the white-footed mice and their counterparts all over the world. It's the species that we allow in when we put that filter in. When we destroy diversity, we are fostering the species that harbor the dangerous pathogens, the ones that jump to humans, the ones that can make us sick. So a lot of that pathogen diversity is irrelevant to our health. The parts that are relevant are generally the ones that occur in the species that thrive when we reduce diversity under disturbance or pollution or degradation. And the rhinos of the world harbor microbes that may be pathogenic, but they are not the ones that we need to worry about. This has been shown over and over, and the Gibb paper, I think, nails that very nicely at a global scale.
Thanks, Rick. I'm going to ask one final question here, and then we'll wrap up. And that question is this. So you showed a figure early on that showed, I think back around the mid to early 1970s, essentially 0 known cases of Lyme, and then it's exploded over the subsequent 50 years. My question is this, do you know much about the origin of Lyme. That is to say, is that pattern a consequence of Borrelia essentially appearing at about that time and perhaps spreading because of the tremendous change in landscape structure through fragmentation and so on, and other factors? Or has it been around for a long time? Have you been able to look into the genome or in any other sort of historical records to say whether Lyme appeared about that time, or has it been around for a very long time and it either was just not detected because it wasn't as common, or whatever? Do you have any
Yeah.
understanding of that, Rick? I think it's just an interesting question because,
Yeah.
you know, COVID-19 looks like something that just popped forth not very
Yeah.
long ago.
Right. Yeah, no, it is a very interesting question. There is a literature on this. It's nothing that I've contributed to personally, but there is strong evidence that Borrelia burgdorferi has existed in its current form for tens of thousands of years. It has been found by a National Geographic expedition in a mummified human from, I think, 6,000 years ago that was found very well preserved in the Swiss Alps on an expedition. And Borrelia burgdorferi DNA could be amplified. This guy suffered from Lyme disease 6,000 years ago. By the genomics work, it's evident that Borrelia existed in Europe before it came to North America, and how it got here, we don't know, but it's been probably 50 or 60,000 years that it's been present in North America. So it's been around. There's evidence from some of the Native American oral tradition of what sounds to many people like Lyme disease occurring in ancestral populations. DNA of Borrelia has been found in museum specimens of white-footed mice that were captured in the 19-teens. So it's been around, which doesn't answer the question of why it emerged when it did, but it does debunk some conspiracy theories. I don't know how many of you have heard about this, that Plum Island, this CIA and I guess US Army installation on Long Island, highly secret, top security area, is been proposed as the location where US government scientists created Borrelia burgdorferi in the lab and accidentally let it escape. So we may scoff at, I mean, there's no evidence for this, and it's false. It's a conspiracy theory. But certain politicians, a member of Congress from New Jersey, wrote a bill that was passed that instructed the CDC to investigate the veracity of this claim that Borrelia was produced as a bioweapon and accidentally escaped. And that is false on its face based on the evidence of its existence for far longer than there has been a United States government or the CIA or the ability to produce a new bacterium. And frankly, it would be one of the dumbest bioweapons you could possibly produce if you think about it as compared to other potentially much more lethal, much more transmissible, much more portable things if you really wanted to hurt or kill a lot of people, which I don't, but others appear to. So it's been around for a long time, Jim, is the bottom line.
Yeah. Fabulous, Rick. Well, we could go on. There's obviously a lot of interest. There's some additional questions here that I didn't answer. If you wish to respond to them, I think you can see that, Rick. I think there was one here about changing virulence, whether there's any evidence for that. But I think we'll pull this to a close. Wonderful presentation. It was so clear, so informative, exactly what I knew I would get from you. I really very much appreciate it. We all do. And we'll be in touch independently just through our normal communication and friendship, Rick. But thank you so much
Sounds
for
good.
the contribution. I know all the others joined me, and it was extremely informative and really a great treat on this Thursday. So thanks so much, and goodbye,
Thank
everyone.
you all.
Thank you so
Thank
much.
you for your attention. Thank you for the great questions. Wonderful to see your smiling faces on the screen. And I can be found and emailed if there are any other follow-up questions. I would be delighted to engage in dialogue with any of you. CaryInstitute.org, you can find me.
Thank you. It was wonderful.
Thank you, everyone.
Thank you, Rick. Goodbye, everyone. We'll see you next week for Marm, Kilpatrick, and Lyme disease and COVID-19. That will be our last lecture. Goodbye, everyone.
Bye-bye.